Thanks so much for the opportunity to present at the BioNano Symposium in this virtual format. I'm excited to share with you our clinical genetic study of a Mexican kindred with an ATXN10 repeat expansion and tell you more about how we use optical mapping and no-AMP targeted sequencing, which supports our understanding of clinical molecular genetics of repeat expansion disorders. This slide gives you some background about the clinical presentation of spinocerebellar ataxia type 10. It's a rare autosomal dominant disorder, presenting first with gait problems, followed by disturbance of speech and eye movements, and some patients develop dementia and mood abnormalities. In addition, seizures are a common feature and usually occur several years after the onset of symptoms. Average age at onset is 34 years, and average disease duration is about 13 years. Only symptomatic treatment like anti-seizure therapy is available to date. Interestingly, SCA10 cases have been primarily found in Latin American countries and are basically non-existent outside of Latin America, except for single cases that were reported in the US, China, and Japan. It's hypothesized that a founder mutation likely arose in an ancestral Native American population when migrated from East Central Asia across the Bering Bridge to North America, and then spread throughout the Americas. The map shows you the prevalence for autosomal dominant SCAs in North and South America, and the percentage of SCA10 of the total SCA population, which is between 5 and 45%, and SCA10 is basically absent in the US and Canada. The pathological hallmark of SCAs are GABAergic Purkinje cell loss and cerebellar atrophy. This is illustrated in this MRI, and you see that circled in yellow. And on the right, you see the neuropathological features from a post-mortem brain from a SCA10 case, and which is predominantly Purkinje cell loss and a reduction in volume of the molecular cell layer. The Calbindin staining confirms the Purkinje cell loss, and the remaining Purkinje cells are dysmorphic with marked reduced dendrites in the molecular layer. And you can see that in the panel E, and it's indicated by the yellow arrow. So now the genetics of SCA10 is based on an expansion of an intronic pentanucleotide repeat in the ATXN10 gene, and was first described by Dr. Ashizawa's group in 2000. The normal repeat length is 10 to 32 repeats, whereas intermediate repeats are 280 to 850 repeats in length. And we see full penetrance of the disease over 850 repeats. That means you need to detect expansions that are between 4.2 and 22 kilobases in length, which can be quite challenging technically. So in this study, I want to tell you about how we use optical mapping through BioNano and PacBio no-AMP targeted sequencing to resolve this ATXN10 repeat expansion. We wanted to analyze DNA samples from affected and unaffected individuals from a SCA10 family and apply no-AMP targeted sequencing and optical mapping from BioNano genomics. Clinically, we wanted to better understand phenotype-genotype correlations. For example, how does repeat length affect the clinical phenotype? How does the repeat composition affect the clinical phenotype? And can we assess somatic mosaicism of the repeat expansion? So as you know, Southern blot analysis can only roughly estimate repeat length and size of a DNA fragment containing these repeat expansions. And while more recently developed repeat prime PCR can help us with the repeat composition, it really cannot resolve the complete repeat expansion, in this case of ATXN10, and only reads about 1 to 1.5 kilobases at the 5' end. And as you just learned, we need to sequence through several kilobases, up to 22 kilobases. So the exact size and repeat composition cannot be detected with current diagnostic methods, but next generation technologies will allow us, or already allowing us, to solve these challenges. So this is the 3-generation pedigree from our SCA10 family that we have been following for many years. And you see black and red symbols, which indicate affected cases, and the dots indicate mutation carriers. The proband in 3.8 originally presented in the clinic, in our clinic, with Parkinson's disease about 15 years ago. And we did SCA diagnostic panel for him, and also his affected brother, who's actually affected with typical SCA10 and not Parkinson's disease. And we found the ATXN10 expansion. But we were puzzled at the time about the clinical presentation of Parkinson's and SCA10 in the same family. So to learn more about this repeat expansion and to understand the underlying clinical differences, we first used high-resolution optical mapping. In brief, this technology relies on fluorescently labeled ultra-high molecular weight DNA, which is then run on a flow cell with nanochannels and imaged. And those images are analyzed and assembled in a genomic map, and we can detect structural variants. So here are 2 examples. In this case, parent-child, as you can see in the partial pedigree on the right. And in individual 4.3, we found a 6.2 kilobase expanded allele. And in 4.7, which is the daughter, we see a much larger expansion of about 13 kilobases, with a number of smaller expansions indicated by the distribution of the green signal in the histogram below. The distribution of these alleles are also shown in the box plot on the right, which we think is reflecting somatic mosaicism and not technical errors. The second technology we applied was no-amp targeted sequencing, with the goal not only to size the repeat, but also to resolve the repeat composition. On the left, you see the standard workflow for no-amp sequencing by PacBio. And in the middle and right, you have examples on how these data can look like and can be displayed. So to explore the repeat structure, we have waterfall plots. And on the y-axis, you can see the number of reads. And on the x-axis, you can see the length of the repeat expansions in base pairs. To examine the repeat characteristics, histograms are very useful and allow for the confirmation of the allele sizes for the normal and the expanded allele, and can show the extent of mosaicism of the expanded allele. So here are some examples for no-amp targeted sequencing. Since we can sequence through the entire repeat of several kilobases, we can also resolve the genomic composition. And interestingly, we found 2 different repeat patterns. On the left, in samples 4, 5, and 6, we have a biphasic. 4 and 5 are 2 different repeat patterns. And on the right, in samples 4, 5, and 6, we have a biphasic. 4 and 5 are 2 different repeat patterns. And on the right, in samples 4, 5, and 6, we have a biphasic. 4 and 5 are 2 different repeat patterns. And on the right, in samples 4, 5, and 6, we have a biphasic. 4 and 5 are 2 different repeat patterns. And on the right, in samples 4, 5, and 6, we have a biphasic. 4 and 5 are 2 different repeat patterns. And on the right, in samples 4, 5, and 6, we have a biphasic. 4 and 5 are 2 different repeat patterns. And on the right, in samples 4, 5, and 6, we have a biphasic. 4 and 5 are 2 different repeat patterns. And on the right, in samples 4, 5, and 6, we have a biphasic. 4 and 5 are 2 different repeat patterns. And on the right, in samples 4, 5, and 6, we have a are represented in gray, together with ATTCC repeats that are represented in orange. And then samples 3, 4, 4, 7, and 8 show a pure ATTCT expansion indicated in gray. Interestingly, again, sample 4, 7 shows a very large variability of the repeat. And when we do the counting, we can count 66 ATTCT repeats up to 2,780 ATTCT repeats, similar to the data from the optical mapping. We were intrigued by this somatic variability and wanted to see if we can also show this at the cellular level. So it's known that the spliced introns of the ATXN10 repeat can accumulate in the nuclei and can be visualized using in situ hybridization. Here we used a custom designed RNA scope hybridization probe for the ATTCT repeat. In panels A through C, we depict a positive control for the human mRNA. D to F shows a healthy control, skin fibroblasts without the ATXN10 repeat expansion as a negative control. And I want to point your attention to panel G through I, which shows skin fibroblasts from the same individual, 4.7 of the pedigree. And that again shows the highly variable repeat expansion. So what we were doing is we were counting all the DAPI cells, and we have 7 cells that show a positive signal that you see in H with the yellow arrow. And there are 4 cells that are completely negative and don't show the RNA foci or the label, which are in red in panel I. So presumably the long expansions lead to prominent aggregates in the cells, whereas the shorter repeats show only small puncta or no aggregation of the repeat RNA at all. So in conclusion, the CRISPR-Cas9 or no-M smart sequencing technology combined with optical mapping allows for accurate sizing of the repeat expansion, can resolve the entire repeat expansion, and establish somatic mosaicism. Exact repeat sizing and composition is critical for clinical practice and genetic counseling, and really can influence disease penetrance, clinical symptoms, and consequently disease trajectory. Here, I'm at the end of my talk, and I would like to acknowledge my collaborator and, of course, the family who is participating in this research. In particular, there is Javier Jimenez, our neurologist from Guadalajara, and then members of the Schuele lab, Joss Palafox, Faria Zafar, and Ani Torres. I'm very grateful for the group at PacBio with Yu Chi and Janet, and then the folks at BioNano, Carl Hong, Joyce Lee, and Jill Lay for helping us generate those data. And last but not least, I'd like to acknowledge Dr. Ashizawa, who is a long-standing collaborator and SCA expert. Thank you so much for watching. And if you have comments or questions, please contact me, follow us on Twitter, or visit our website. Thank you so much.